This is week 12, Molecular Genetics and Human Disease, part 1.
The learning objectives for this week are to describe conditions resulting from mutations in mitochondrial DNA, to describe conditions resulting from receptors including hypercholesteremia, to describe conditions resulting from transport defects including cystic fibrosis, to describe conditions resulting from production of abnormal structural proteins including the muscular dystrophies, and to describe diseases resulting from expansion of unstable repeat sequences, including Huntington disease and Fragile X.
Let's start off by looking at mitochondrial diseases. Mitochondrial genes encode proteins that participate in protein synthesis and energy production. Remember, mitochondria replicate independently and they perform aerobic respiration, which consists of Krebs cycle and oxidative phosphorylation. 37 genes total make up the mitochondrial genes. There are 24 of those genes that encode for RNA molecules, 22 for transfer RNAs, and 2 for ribosomal RNAs. Remember that the transfer RNAs and the ribosomal RNAs work with the messenger RNAs for protein synthesis. The other 13 mitochondrial genes encode proteins that function in cellular respiration and the production of ATP. Just to refresh everybody's memory, here we can see the mitochondria, which are out in the cytoplasm. Remember your cellular biology that the more mitochondria that you have in a specific area, this means that there is more activity that is taking place. As I mentioned, they do replicate independently. Remember that when we're looking at the ribosomal subunits, they are not the same as other eukaryotic cells. They are more like prokaryotes. If we take a look at the mitochondrial DNA, here you have the 37 genes in a circular formation, and you have the double-layered membrane that's present. Now let's go back to the inheritance of a mitochondrial disease. Let's remember that the mitochondria are in the cytoplasm. When fertilization takes place, it is only the nucleus of the sperm that makes up the zygote. And therefore, any cytoplasmic the mitochondria in the cytoplasm are going to come from the female. So therefore, if a female has a mitochondrial disease, all of her offspring will be affected. If a male has a mitochondrial disease, he will not pass on the disease to his offspring. A class of diseases that are referred to as the mitochondrial myopathies are often lumped together, even though they have specific names, and they're referred to as mitochondrial disease. Their symptoms arise from the tissues whose cells normally have many mitochondria, such as skeletal muscle. And some of the symptoms that you see with these diseases are that individuals have a lot of fatigue, they're weak, they have flaccid muscles, and intolerance to exercise. If we look closely at the skeletal muscle fibers and we look under the light microscope after staining at the mitochondria, they're abnormal. And the skeletal muscle fibers appear red and ragged. This is a slide showing you pointing out red and ragged fibers. Mitochondrial diseases may also result from mutations in nuclear genes that encode proteins that are essential for mitochondrial function. A mutation in a mitochondrial gene that encodes a transfer RNA or a ribosomal RNA can be devastating because it impairs the mitochondrial's ability to manufacture proteins. About 1 out of every 200 individuals has a mutation in a mitochondrial gene that could cause a disease. These are rare diseases. They affect about 1 in 6,500 individuals. And this is also due to the fact of selection that takes place during the egg formation. Since a disruption of energy acquisitions and the production of ATP can be lethal to an oocyte. So what if we could replace the mitochondria with healthy ones from a donor? Bioethically, there have been concerns because this would mean that there were three parents involved with the production of one child. So, two ways that have been looked at with animal studies, and there are other ways also that are being looked at, is number one, to transfer the male and female pronuclei from an in vitro fertilized ovum into an oocyte from a donor of a healthy woman that has had the nucleus removed so that the pronuclei share a cytoplasm with a donor mitochondria. Or, number two, transfer the female pronucleus into a donated oocyte with its nucleus removed and then fertilize the cell in vitro. Let's go back and review heteroplasmy. This is where a mutation is in some mitochondrial chromosomes but not in others. And then remember that at each cell division, the mitochondria are distributed at random into daughter cells. Over time, the chromosomes within a mitochondrium tend to be all mutant or all wild type for any particular gene. But different mitochondria can have different alleles predominating. Now then what happens is as an oocyte matures, the number of mitochondria drops from 100,000 to 100 or so. And women who are heteroplasmic for a mutation can produce an oocyte that has mostly mitochondria that are of the wild type, some that have mostly have the mutation, or anything in between. A woman who does not have a mitochondrial disease can then pass it on, and this makes it very complex and unpredictable. So most severe mitochondrial illnesses are heteroplasmic. Expressivity will vary among the siblings, and the severity of symptoms reflects which tissues have cells whose mitochondria bear the mutation. Often, heteroplastic mitochondrial diseases do not produce symptoms until adulthood because it takes many cell divisions and therefore years for a cell to receive enough mitochondria bearing mutant alleles to cause symptoms. I want to look at one disease here called mitochondrial myopathy, encephalopathy, lactic acidosis syndrome. And I want to look at a nurse who was affected with this disorder. And so we look at this nurse, a once active and articulate individual. In her 40s, she gradually began to slow down at work. She heard a buzzing in her ears and developed difficulty talking and walking. Then her memory began to fade in and out. She became lost easily in familiar places, and her conversation made no sense. Her condition worsened, and she developed diabetes, seizures, and pneumonia, and became deaf and demented. Finally, she was diagnosed with MELAS, which stands for mitochondrial myopathy, encephalopathy, lactic acidosis syndrome. Her muscle cells had the telltale red ragged fibers, and she died. She had a daughter and a son, and because this is a mitochondrial disease, both her son and daughter will likely develop the condition in the future. One athlete who was afflicted with a mitochondrial myopathy was Greg LeMond. He was a cyclist and he was a three-time winner of the Tour de France. You can read about him in the link that I have provided here.
This slide is showing you how mutations can cause disease, and included in this chart are some of the diseases that we're going to be looking at this week: cystic fibrosis, Duchenne muscular dystrophy, familial hypercholesterolemia, and Huntington disease. Mutations are classified by whether they remove, alter, or add function, or by how they structurally alter DNA. The same single gene disease can result from different types of mutations. If we look at this table, it summarizes types of mutations using an analogy to an English sentence. We can see the English sentence is one big fly had one red eye. A point mutation is a change in a single DNA base. A transition is a point mutation that replaces a purine with a purine, such as A to G or G to A, or a pyrimidine with a pyrimidine, such as C to T or T to C. A transversion is a point mutation that replaces a purine with a pyrimidine or vice versa, A or G to T or C. Addition or deletion of a single DNA base is also considered a point mutation. A point mutation can have any of several consequences, or it may have no obvious effect at all on the phenotype, acting as a silent mutation. A point mutation that changes a codon that normally specifies a particular amino acid into one that codes for a different amino acid is called a missense mutation. If the substituted amino acid alters the protein's conformation significantly or occurs at a site critical to its function, signs or symptoms of a disease or an observable variant of a trait may result. About 1/3 of missense mutations actually harm health. Familial hypercholesteremia represents an autosomal dominant disorder that has incomplete penetrance. The phenotypes are related to the amount of receptor cells on the liver cells, the hepatic cells, that take up the low-density lipoprotein LDL form of cholesterol from the bloodstream. Deficient LDL receptors cause cholesterol to accumulate in the blood. This can lead to premature heart disease. A rare homozygous dominant individual would die at a young age from coronary disease. Heterozygous individuals suffer heart disease in early adulthood. If we look at familial hypercholesteremia, it's caused by mutation in the LDL receptor gene LDLR. The signs and symptoms are high blood cholesterol and early heart disease. Physical stigmata include xanthomas or cholesterol deposits in the skin and tendons, premature arcus cornea, which are deposits of cholesterol around the periphery of the cornea, and it is one of the most common single gene disorders, occurring about 1 in 500 individuals. It's important to identify early for cholesterol-lowering therapies to prevent coronary artery disease and to identify other family members at risk. Homozygotes will ultimately require liver transplantation. If we look at this slide, we can see that a missense mutation in the gene encoding the low-density lipoprotein LDL receptor that is abnormal in familial hypercholesteremia. And if we look at A, and if we look at B, and we look at C, in A we see that it bears a missense mutation. A cysteine substitutes for a tyrosine, binding the receptor enough to impair its function. The short LDL receptor in B results from a nonsense mutation in which a stop codon replaces a tryptophan codon, and in C, a 4-base insertion adds an amino acid that then alters the repeating frame. Plus and minus indicate electrical charges on the amino acids. Cystic fibrosis is an autosomal recessive disorder. There are more than 2,000 variants currently known of the CF transmembrane regulator gene, CFTR, which encodes the chloride channel protein. The carrier rate of 1 out of 25 is found in the white population, and 1 out of 2,500 white births results in cystic fibrosis. It is not as common in the Asian population nor in the African American population in the United States. It is actually the most common fatal autosomal recessive disease in white children. Today, we test pregnant women for the more common mutations to see if they are carriers, and in most states it's part of the newborn screening panel. When cystic fibrosis was recognized in 1938, the life expectancy was about 5 years. However, today the median survival is about 40 years, with many patients living longer. The major clinical consequences are the production of abnormally thickened mucus secretions which are found in the lungs and in the digestive system. This is because the cystic fibrosis gene affects the ion channels that control chloride movement out of cells. And we see that the thick, sticky mucus, when it accumulates in the lungs, creates an environment which is hospitable to certain bacteria that are common in cystic fibrosis patients but not seen in healthy lungs. A clogged pancreas will prevent the digestive enzymes from reaching the small intestine for digestion, impairing nutrient absorption. And a child with cystic fibrosis has trouble breathing and maintaining weight. And in many cases, babies will present themselves to an emergency department or to the pediatrician with failure to thrive. The major diagnostic feature of increased sweat, sodium, and chloride concentrations is utilized in doing what is called a sweat test. In fact, back in the 1970s when I first became a genetic counselor and worked in a CF clinic, one of the things that the doctors would say is, have you kissed your child today? Due to the fact that they have that increased taste of the salt. In most patients, the diagnosis is initially based on the clinical pulmonary and/or pancreatic findings and on the elevated level of sweat chloride. Approximately 5 to 10% of patients with CF are pancreatic sufficient and have a much better prognosis since their nutrition is not impaired. 10 to 20% of newborns are born with meconium ileus, which is a lower intestinal tract obstruction. Females who have the disorder are less fertile, and more than 90% of males are infertile because they lack the vas deferens, and they have a condition called congenital bilateral absence of the vas deferens, or CBAVD. If we look at some of the drugs that are now being used to treat cystic fibrosis, we can see that their modes of actions differ depending upon what the specific defect an individual has. For example, we can correct the misfolded CFTR proteins. We can restore liquid on airway surfaces. We can break up mucus. We can improve nutrition. We can fight inflammation and infection. And we're able to open up the ion channels.